animal study, it was a cross-sectional convenient random sampling of slaughtered animals. We did it in two parts. In the first part we collected 997 cervical lymph nodes between the year 2008 and 2009. A questionnaire was administered to the owners of the samples of the pigs that we sampled. The second part, we did this in 2011. We sampled 150 mesenteric lymph nodes. The human case study was a cross-sectional study done at the referral hospital in Mubende district in 2011. The inclusion criteria was an individual who came into the hospital with cervical lymphadenitis or a cough that had persisted for at least 2 weeks. In this regard, we sampled 344 patients from the outpatient department, the TB ward, the pediatric lab ward, and the HIV and AIDS department. Questionnaires were administered to every person that we sampled, but also we retrieved their HIV status from their case file if this had been done. Then the data assembly was done in Excel. We assembled the data in Excel. This was validated. The data analysis, epidemiological data analysis, was done in Stata. Molecular analysis was done in bionumerics and Miro VNTR Plus with the help of the online resources of the STVIT2 database that is hosted by the Pasteur Institute in France. Databases included the household database for the environmental study, the pig owners database from the animal study, and the patients database from the human study, but of course a laboratory database for all the data that we got from the laboratories. Laboratory work involved direct acid fast staining. Now we had to do the direct acid fast staining because in Mubende this is the diagnostic that they use before treatment starts if you have TB. And anything else that we shall see in terms of culture growth, in terms of molecular work, these are the unknowns. That the decontamination was done for all the 3 studies, and acid fast and microscopy was done for all the 3 studies. DNA extraction was done for all the 3 studies. Species identification was done for all the 3 studies, but molecular genotyping and characterization was done for only the animal and the human study. Of course, to carry out this work you need to get ethical clearance, and we got it. It was pretty challenging, especially being a veterinarian and you have to have access to human samples. It took us close to 8 months to actually get ethical clearance to carry out this work. We did get the ethical clearance from the Uganda National Council of Science and Technology, but also because we had to access government archive, including meteorological data and all facets of data that we had to overlay in order to give a complete epidemiological picture. We had to get special permission from the Office of the President. And after all that, we produced 6 articles, and we shall now go through the results of results and discussion. We'll start with the environmental case study. We found that 15% of the samples that we cultured from the environment actually had mycobacteria in them based on the acid fast test. And soil and water had the highest number of mycobacteria isolated in them, but you also see that the animal waste contains mycobacteria. That means that the animals are playing a key part in contaminating the environment and the environment. We found more slow-growing mycobacteria, and by slow-growing mycobacteria, these are the ones that are documented to be more pathogenic to animal and human. We found more of these in the household water, that water that they use for washing dishes, for showering, for drinking, and all that, the whole host of things that you would use, domestic water for. We see that the isolation of mycobacteria was more, or almost 35% of all the mycobacteria isolated was recovered from at the household level. 40% of the mycobacteria isolated was recovered from at the household level. 40% of the mycobacteria isolated was recovered from at the household level. 40% of the mycobacteria isolated was recovered from at the household level. 40% of the mycobacteria isolated was recovered from at the household level. 40% of the mycobacteria isolated was recovered from at the household level. 40% of the mycobacteria isolated was recovered from at the household level. 40% of the mycobacteria isolated was recovered from at the household level. 40% of the mycobacteria isolated was recovered from at the household level. 40% of the mycobacteria isolated was recovered from at the household level. 40% of the mycobacteria isolated was recovered from at the household level. 40% of the mycobacteria isolated was recovered from at the household level. 40% of the mycobacteria isolated was recovered from at the household level. 40% of the mycobacteria isolated was recovered from at the h and potentially pathogenic mycobacteria, that for example those that belong to Mycobacterium avium complex were recovered. And of course Mycobacterium simie, this has been documented to cause a lot of disseminated infections in HIV patients. The slow-growing mycobacteria were also isolated most in unboiled water. Now remember that the attitudes of these pastoralists are that they know that water could be the source of these infections, but they're not willing to treat it. And here we are actually finding more slow-growing mycobacteria that is potentially pathogenic to them and the animals in these untreated water. But also households that kept cattle, we found more mycobacteria from their households, basically due to the fact that they share water sources with these animals. And we believe that when the valley dams are much more contaminated, and due to animal sharing and fecal droppings, and I think you can see that this gentleman was collecting water together with the animals, and probably the water is going to use at home, would explain why we found a lot of mycobacteria in the household water. But we also found quite a lot of mycobacteria in valley dams, and we believe that we also found that there was a temporal association with precipitation. What do I mean with this? Basically, as the rains, the precipitation increases, there was also an increase with mycobacteria isolated, both slow growing and fast and rapid growing. But the increase was much more evident in the slow-growing mycobacteria, the ones that are potentially pathogenic. But beyond a certain point, the saturation kicks in, and the dilution factor kicks in also. At the amount that we isolated beyond a certain amount of rainfall in this area seemed to dilute what we got. Therefore, so this simply means that during the rainy seasons, the stormwater from the hilly areas washes down heavily organic and organically rich soils and everything into the valley dams. This becomes a rich environment for non-tuberculous mycobacteria to grow. Unfortunately, these collecting areas are the same areas these pastoralists go to get water for home use, but also take their animals for drinking water during the dry seasons. And like I said, these people seem to be caught between a rock and a hard place, because they need this water on a personal level, but their animals also need it. But the same water is actually containing more mycobacteria during or after the rainy seasons. And it's also expected as the dry seasons come in and the water starts evaporating, it should be the concentration of mycobacteria should start picking up, given that it works on a dilution factor. As the water evaporates, it leaves that behind this high concentrate of muddy water that is expected to have high concentration of mycobacteria. So these people are actually on both ends of the season, they are getting it, in abundance. This study, the results from the animal case study, we isolated non-tuberculous mycobacteria from slaughtered pigs as well. We found that 39% of the cervical lymph nodes that we cultured actually had non-tuberculous mycobacteria, and 31% of those actually did not have lesions. That means on routine inspection, these would have been missed, and depending on how well cooked your meat would be, as a consumer, as a person living in Mubende, depending on how well the meat would be cooked, you probably would be exposed to these non-tuberculous mycobacteria. Majority of them actually were the pathogenic type, the M. avium. 19% of them were M. avium, and majority of them, or when we did the speciation, we found that M. avium subspecies hominis was the most prevalent. This is known to cause infections in humans, it's known to cause infections in pigs, but also Mycobacterium avium subspecies avium is known to cause infection in poultry, but has also been documented to cause infection in humans, especially those who are immunely compromised. This isolation was much higher than what has been documented in Nigeria and Switzerland, and we believe that this is due to the difference in animal management. I mean, pigs in these areas are left to roam around to fend for themselves and look for food. So when we ran a mixed logistic model, we found that pigs that were from households that got water from valley dams were 2 to 3 times more likely to have non-tuberculous mycobacteria within them. But also, when we analyzed pigs that were from households that practiced free-range type of, that means they left their pigs to go around to look for food, as compared to those who housed them, we found that those pigs that were left to go roaming actually had 3 times more non-tuberculous mycobacteria than those that were housed. Then when we compare the environment and the pigs then seem to be terrestrial filter feeders. They accumulate non-tuberculous mycobacteria as they go around fending for looking for food. They are accumulating mycobacteria from the environment, just like the aquatic filter feeders would do. They are taking in water and they take in the particles for feeding purposes, basically. But in the end, they concentrate whatever microorganisms are in the water. The pigs are doing almost the same in the environment in Mubende, and this is exactly what we mean. If we compare the summary of species that were isolated from the environment and those that were isolated from the pigs, we take an example of Mycobacterium senusei. We see that in environment we isolated only one Mycobacterium senusei, and this is understandable given the dilution factor that we just talked about, that is controlled by the precipitation. We've isolated 24 from the animals, so that means the animals have more than what is in the environment. The M. terry also we isolated 1, but isolated 9 in pigs. The same thing. So there's a direct relationship between, in terms of species that are prevalent in the environment and those that are in the pigs, but they are much more amplified in the pigs. And this, we think, is because the pigs have to go around fending for themselves, looking for water and food, and in the end they end up accumulating this whole host of and of course, the pigs that are housed indoors, for example in Europe, in Holland, they have a whole host of, they have different types of risk factors. In those areas they actually isolate a limited variety of mycobacteria, but in higher numbers, given that they are housed intensively, so they are definitely exposed to the same type. These ones, on the other hand, have a wide variety of mycobacteria isolated with them, so definitely it is a component of the management system that is responsible for the wide variety, but also the fact that pigs are concentrating it as they look for food.